All right, so your first thing, your Unit 4 Frayer cards are in the drawer. So we didn't do the whole changeover of the organization. If you remember how to do it and you want to try to do it, you can. You'll attach Unit 3, which doesn't have a whole lot. You'll attach that to your ring and then take Unit 4 off. If you don't want to do that, just keep Unit 4 separate for now, and we'll do it when I get back. So my concern for today is that you have all the first 4 Frayer cards done. So we're going to go through each of the 4 Frayer cards that you're going to do today. So the first one's going to be plasma membrane. If you need to get the Frayer cards, hit pause on the video, go get them, and then start the video again once you are ready with your materials. So again, Unit 4 Frayer cards in the drawer. George, just take Derek's cards that are in the drawer because those are already set up. Okay. So definition of plasma membrane. It is a structure found on the outside of cells that controls transport. So it is all the way around the outside of the cell. Every cell has a plasma membrane, every single cell. Plant cell, animal cell, prokaryotic bacterial cell, they all have plasma membranes. And their job is, yes, it protects the outside of the cell, but it does that by controlling what comes in and what can come out. So it's like a border around the outside. So when we looked at those examples in the Nearpod, it's like the fence, right? So it keeps the dog in, it keeps people that don't live there out, but people can move in and out through the cell membrane. A characteristic about plasma membranes, that should say some stuff gets in, not some stuff gets I. All right, plasma membranes are semi-permeable. We're going to learn the word permeable later, but semi-permeable is how we describe them. So semi, a little bit, permeable, things can get in and out. That we have our own Frayer card for that word. Some stuff gets in based on need. Some, yeah, there we go. Oh, there we go. Some stuff gets in based on need. If it's good, it gets in. If it's bad, it doesn't. Other names for it, we typically call it the cell membrane because we do, right? It's the cell membrane, but plasma membrane is like the more official sciencey name for it. It's also called the phospholipid bilayer. So many words, so many syllables, but that really tells you a lot about its structure. We have our own Frayer card for phospholipid bilayer to break that up. Our non-examples is a cell wall, right? They both surround cells, cell membranes and cell walls. They do both surround cells, but they're not exactly the same thing. They have slightly different functions and very different structures. So we're going to use that for a non-example because they're not identical. For the drawing of the plasma membrane, all I want you to draw for now is just a couple of phospholipid heads and tails like that. Okay, just put like 3 of them, top and bottom, 2 layers. We're going to put 1 protein channel in there. So I think of it like a hot dog bun, right? So I'm drawing like if we're looking down at a hot dog bun. Couple more phospholipids. And then I do another solid one with a piece sticking off the top, whatever shape you want to kind of make it look like an antenna. And then some more phospholipids. Every phospholipid head is supposed to have 2 tails. Sometimes I mess up when I'm drawing it, but it's supposed to have that. So pause the video if you need to go through what the extra, or what the drawing, the characteristics, the non-examples, examples, or definition. Pause, rewind. I'm going to move ahead to the next term. Our next term on here. There it is. Phospholipid bilayer. So the definition of phospholipid bilayer is a structure made of 2 layers of phospholipids. It's a synonym for cell membrane. It's a synonym for plasma membrane, but we're going to be specific with the name. Bilayer, 2 layers. Bicycle, 2 wheels. Bilayer, 2 layers. Phospholipid is the actual phospholipid that we have. Phospholipid. That's a phospholipid. It has a head and a tail, a double tail, head and a tail. So it's made of 2 layers of those. Characteristics, specifically the phospholipid bilayer, you need to know those heads are hydrophilic and the tails are hydrophobic. Hydrophilic means that it loves water. Hydro, water. Philic, like Philadelphia, city of brotherly love. Philic is the Latin word for like loves or gets along with or will be with. The tails down here, those are hydrophobic. You've probably heard the word phobia before. They hate water. So I bet you could think of things that are hydrophobic, things that won't mix with water. Think back to our lava lamps we made at the start of the year, right? That oil did not mix with water. It stayed separate. So because our cell membrane, because our phospholipid bilayer has this outer side, it could float in the water, right? The phospholipid head is fine floating around in the water. But the tail does not mix with water. That keeps it from dissolving. Because you have 70% water in your body, your cells, you'd melt like the Wicked Witch of the West because your cells would dissolve. So the makeup of the cell membrane, since those hydrophilic heads go all the way on the outside and all the way on the inside, I'm going to try to do a good circle. Yes, almost. And the tails are all in here. It keeps the cell separated from its watery environment because the tails are on the inside, the heads are there. So it makes that good barrier. So those are 2 snuck in vocab words there. Examples, again, just the synonyms for it, right? It's a plasma membrane, it's a cell membrane. Other words for the same thing. Phospholipid bilayer just more specifically describes it as what it is. It does allow small molecules to pass through. So even though these phospholipids are all right up next to each other, right? All those heads are super, super, super connected. Super, 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 super connected. I even draw them overlapping. That's not exactly right. And the tails are in the middle. If it's a really teeny tiny molecule like oxygen, that can just go right through the membrane. If it's bigger, it's not going to be able to do that. For the drawing of the phospholipid bilayer, you can, if you want to, redraw it, or you could just say same as the cell membrane. So if you want to redraw it, go back to your cell membrane card. If you don't want to redraw it, just say same as, or plasma, sorry, same as plasma membrane. They are the same visual because they're synonyms of the same word. So if you need more time on this card, hit the pause button. If you don't, we're going to move on to our next term, carrier protein. So a cell membrane doesn't only have phospholipids in it. It also has proteins. It also has carbohydrates. So I'm going to go back to this image here. This is the carrier protein that we're talking about. So for your drawing, you are going to redraw it, but we're going to focus just on that part. Carrier proteins are proteins that are embedded. Embedded is just a fancy word for stuck in. Not loving this giant pen I have here. There we go. They are stuck in the plasma membrane, and that allows larger molecules in and out. So if we think of, and it thinks that it's the same word I don't want you to use, but like if we think of the wall of the classroom, right? If we're in the classroom, that wall separates us from the hallway. But
good morning, teachers and staff.
Hold.
Breakfast is now ready.
Sorry. So the wall in our classroom separates us from the hallway, but we can still hear the noise, right? That sound can get through the wall. But if a person wanted to come into the room, the person is too big. They can't get through the wall like the sound can. They have to go in and out of the door, right? You have to open the door to get in and out. Light can't get through that wall, but light can get through the window, right? There's different openings. The vents in the ceiling, the air vents in the ceiling, they can get in and out through, air can get in and out of there. We can't. So there's different membranes, different proteins in the membrane that allow different things in and out of the cell. Characteristics of the proteins, some are specific to certain molecules. So like very, very specific, like only this exact type of sugar can make its way through this particular channel or opening or pore or doorway, whatever you want to think about it, in the membrane. Some will just let anything in. Other names for them, transport proteins and protein channels. We're going to build a model of a cell membrane and you're going to see the kind of little bit of differences in the shape. The idea of a carrier protein is it gets things from one side to the other of the cell membrane. Our non-example would be a marker protein. The marker protein, that's the one that has the carbohydrates on it. So the carbohydrate is attached to the like protein that's just stuck in there. So I'm going to go back to the other drawing that I did. Okay, so here we go. For the carrier protein, you're going to redraw this with an arrow. It's this protein here. It's the one that allows things in and out. This one is a marker protein. This part up here is the carbohydrate, but this thing that's stuck in the membrane, that's our marker protein that it attaches to. So you're going to draw this picture again. It's the same. I know you're drawing it again, but I really want you to get comfortable with all the parts of the membrane, all the phospholipids, the actual carrier protein, and then the opening that's in between it that allows things to get in and out of the cell. Again, if you need to pause the video, go ahead and pause it. I'm going to move on to the next definition. The last definition for today is permeable. There's no drawing for this really. I might try to like throw something in, but there's not like an actual drawing. So permeable is an ability something has. Permeable allows molecules to pass through. So a paper towel, right? Water can get through the paper towel. Water cannot get through glass. So allows molecules to pass through. We're going to talk about it in regard to the cell membrane. What is it allowed to get into the cell or out of the cell? What does it actually move through? It's like a sponge. So I said paper towel, right? It absorbs. Things can get into it and get through it if something is permeable. The cell membrane is semi-permeable. It lets some things in sometimes. Water, we need water. We don't need too much water. So it lets things in, we said it in the first one, based on need. It gets into the cell based on need. So the cell membrane is semi-permeable. That's a vocab word you're going to want to recognize as we go through the transport unit. There's not really an example because cell membranes are permeable. Cell walls are way more permeable. They let everybody in. They're like, yeah, sure, come on. And then the cell membrane's like, whoa, 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 whoa. We don't need any more of that. So cell membranes are permeable to some things. Again, water, oxygen, carbon dioxide. Yes, we need those to pass through. Sugar, we need those to pass through. But there's a maximum. Sometimes we don't want any more water or the cell might explode. So it kind of like controls its opening ability. Non-examples, these are the antonyms for the word. So non-permeable, impermeable. You're probably going to see impermeable a lot more. So when we're talking about cell membranes, you'll see things like this cell membrane is permeable to water, but impermeable to salt. And you have to, in your head, that means Water can get in, permeable. Salt cannot get in, impermeable. Non-porous, nothing gets absorbed, gets through. Porous and permeable aren't synonyms, but they're similar to each other. So for the drawing of permeable, it's just things moving through. It's the ability of things to move through. So it's a hard thing to draw. So go back if you need to review any of these. If you didn't pause it when you were going through, make sure you have all the definitions, all the drawings. And you can just put these cards either back in the drawer if it's too crowded, or put them up on the hooks. All right, if you have any questions, send me an email.